In today's lecture, we are going to study protein sequencing. So what is meant by protein sequencing? So protein sequencing is a technique in which we have to determine the amino acid sequence of a protein. So all of you know that the proteins are made up of amino acids, and these proteins are building blocks of the system. So to determine what is the sequence of amino acid in the protein is known as protein sequencing. So protein sequencing is a method to understand the structure and function of proteins in living organisms. So by using protein sequencing, we can understand the structure and function of the proteins in living organisms. Then amino acid sequence will determine eventual three-dimensional structure of the protein. So if we know the amino acid sequence of the protein, then from that amino acid sequence, we can determine the three-dimensional structure of the protein. Then protein sequencing gives information regarding which conformation the protein adopts. So from this sequence, we can get to know how the conformation of the protein is. Then from this, we can discover the structure and functions of the proteins in living organisms, and it is an important tool for understanding cellular processes. So if we want to study the cellular processes, then what is important? We should know the structure and function of the protein. So as we are knowing the sequence of the protein, from this sequence, we are determining the structure of the protein, and from the structure of the protein, we can determine the function of the protein in the cellular processes. Then it allows the drugs that target specific metabolic pathways to be invented more easily. So by using protein sequencing, we can find out the target. So from that target, we can design the drug, and we can block any of the pathway, and we can discover the drug. Then the first protein sequencing was achieved by Frederick Sanger in 1953. Then he determined the amino acid sequence of bovine insulin. So first protein sequence has been determined to that of the bovine insulin, and it was determined in the year 1953 by Frederick Sanger. And Sanger got a Nobel Prize in the 1958. So he discovered, or he sequenced the insulin protein in 1953, and he got the Nobel Prize in 1958. Now the methods of protein sequencing. So there are two methods of protein sequencing. First is Sanger's method, and second is Edman's method. So we will study first method, that is the Sanger's method, or we can say Sanger's and Dancil chloride method. So again, this Sanger's method has been given by Frederick Sanger, and he got a Nobel Prize for this method. Then he showed for the first time that amino acids are covalently together by alpha amino and alpha carboxyl group. So he was the first who showed the first time that amino acids are covalently bound together by alpha amino, that means the amino group of the first amino acid, and carboxyl group of the second amino group. So, so this covalent bond holds the two amino acid together. So this was showed first by the Sanger, and for this he got the Nobel Prize. Then he suggested stepwise release and identification of amino acid starting from the N-terminal end. So as we know that that particular polypeptide chain has the two ends. First is C-terminal, or we can say carboxyl terminal, and second one is N-terminal or amino terminal. So what he did, he suggested a stepwise release of the amino acid. So one by one amino acid has been released from the polypeptide chain, and then afterwards identification of the amino acid from the N-terminal end. So one by one the identification of amino acid sequence has been done, and the protein sequence of insulin has been determined. So for this, he used a reagent, that is fluorodinitrobenzene, that is FDB, which is commonly called as Sanger's reagent. So by using this reagent, he carried out the protein sequencing. So what this fluorodinitrobenzene is? So this fluorodinitrobenzene reacts with free amino group of the N-terminus. So whatever the amino acid that is present at the N-terminal of the polypeptide chain, that free amino group will react with this Sanger's reagent. That means fluorodinitrobenzene. So that means we are labeling that particular N-terminal amino acid with this fluorodinitrobenzene or Sanger's reagent. Then next, upon hydrolysis, a yellow colored dinitrophenol, that is DNB derivative of N-terminal amino acid is produced. So after labeling the N-terminal amino acid with the Sanger's reagent, we have to carry out the hydrolysis, and after hydrolysis, a yellow colored dinitrophenol derivative of N-terminal amino acid will get produced. Then the DNB amino acid is identified comparing it with the known standard DNP amino acid by using gel chromatography or different techniques such as high performance liquid chromatography. So this is the principle of Sanger's. So this is the principle of Sanger's method, that is use of this fluorodinitrobenzene reagent for the labeling of N-terminal amino acid of the polypeptide chain, and afterwards has to be carried out, or we can say acid hydrolysis of this labeled amino N-terminal of the polypeptide chain, and afterwards a yellow colored dinitrophenol derivative of terminal amino acid will get produced, and this we have to separate or load into the Dovex 50 column. So column chromatography here we have to carry out, or we can use any of the chromatographic method. So if we load this into that column, then we can get the elution, and from that we can determine each amino acid. So first we will carry out for the first N-terminal amino acid. Then after carrying out this, we will compare it with the standard, and after comparing it with the standard, we can get to know which amino acid was present at the N-terminal chain of the polypeptide chain. So here you can see this 1-fluoro-2,4-dinitrobenzene, that is Sanger's reagent. It is a very reactive towards nucleophilic aromatic substitution. So here you can see this is the 1-fluoro-2,4-dinitrobenzene, that means Sanger's reagent, and it is very reactive towards the nucleophilic aromatic substitution. And this is our polypeptide chain. So whose sequence we have to determine. Then what we will take? We will take the sample and we will add the Sanger's reagent. So after adding this Sanger's reagent, what will happen? This it will be labeled with the Sanger's reagent. So here you can see a HF has been released. This fluorine and hydrogen from the N-terminal amino acid has been released, and this labeling has been happened. So this is the first step in the Sanger's method. Then next, acid hydrolysis will cleave all of the peptide bonds, leaving a mixture of amino acids, only one of which bears 2,4-DNP group. So afterwards, we have to carry out the hydrolysis. So for this, we can use 6 molar HCl at 110 degrees Celsius. So by using this high temperature and or we are using acid over here. So acid hydrolysis, it will leave a mixture of amino acids. So previously we have seen, so these are the amino acids which are present in the polypeptide chain. So upon hydrolysis, what will happen? It will release the amino acid mixture. That means each amino acid will get separated, and it will leave the one amino acid which bears the 2,4-DNP group. That means this N-terminal amino acid is labeled with this Sanger's reagent. So this we will get. Then what we have to carry out? We have to load this into the Dovex 50 column, or we have to carry out the HPLC, and by using this, we can compare the results with the standard, and from that we can get to know which amino acid was present at the N-terminal of the chain. So this is for the first N-terminal amino acid. Then afterwards, we will take again sample, and we will clean that first amino acid, and again we will carry out the same reaction. And likewise, we can get to know the amino acid sequence of the polypeptide chain. So apart from this Sanger's reagent, we can also use densyl chloride. So densyl chloride we can use over here. So again, this densyl chloride is used because it forms an intensively colored derivative. So as the color derivative is formed, we can determine that particular amino acid by using chromatographic technique. Then that can be detected with very high sensitivity than the dinitrophenol compound. So as that of the Sanger's reagent, we get the yellow colored derivative, DNP derivative. So likewise here also we are getting the color derivative. So again this densyl chloride react with an uncharged N-terminal to form sulfonamide derivative, so which is stable under conditions that by hydrolyzer peptide bonds. So again this densyl method for determining the amino acid terminal residue, it is sensitive and powerful. So again samely we are using densyl chloride. So densyl chloride we will add into our sample. Then again our sample will be labeled with this densyl chloride. Then afterwards we have to carry out the hydrolysis, and after hydrolysis the N-terminal amino acid residue which is labeled with the densyl chloride we will get, and other amino acids will get separated. So again we have to carry out the HPLC, and we have to determine which amino acid was present at the N-terminal peptide. Now we will see what are the disadvantages of this Sanger's reagent and densyl chloride method. So first is it cannot be used repeatedly on the same peptide because the peptide is totally degraded in the acid hydrolysis step. So because we have seen there are two steps involved. First is we have to add the Sanger's reagent, and second we have to carry out the acid hydrolysis. So as we are using acid hydrolysis over here, that polypeptide will get totally degraded. So if we want to know the second sequence, that second amino acid from that particular chain, so that time we cannot determine by it by using the same sample. So how we can determine then? Then for this, again, we will require that sample. We will remove that first amino acid and we will carry out the same reaction, and we can get to know the second amino acid sequence from the N-terminal end. Then the next disadvantage of this technique is these dyes are selectively label amine group which is present in the protein. So therefore, it may possible that that can label amine group which is present in the side chains, as well as may give erroneous results. So as we know that the proteins are made up of different polypeptide chains, so it may happen that if the side chain is present or a side chain is linked to that particular polypeptide, then that time it may happen that it may label to the amine group which is present at that particular side chain. So that in that case we can get the false result. So these are the two disadvantages of Sanger's method.